Right, it's the next stage. So we've just done ultrafiltration. We're now going to go on to our selective reabsorption, because what's happened so far is that we've filtered any molecule which is smaller than 69,000 relative molecular mass out of the bloodstream into the nephron. And that's a bit of a problem because that includes all the good stuff like glucose, amino acids, waters and salts. So all of that material, along with the toxins like urea, are now in the nephron. And if we carry on this journey, those materials would be lost out and eventually off to the bladder. So what we need to do is to reabsorb the good stuff in section 2, which is the proximal convoluted tubule. And because we're actually selecting what we're going to reabsorb, this area is all about selective reabsorption. So the next bit of work is, in fact, called selective reabsorption. So this is in the proximal convoluted tubule. Right, so the cells of the proximal convoluted tubule. So what I'm going to draw is the tubule walls kind of blown up big style, so we can look at those cells and how they are adapted to their function. So what we have then are the cells of the proximal convoluted tubule that look like this. So the cells which are on the inside of the nephron have microvilli. On the other side, which is in the contact with the capillaries, are folded, so they have a large surface area, but the microvilli has a very large surface area. I'll explain why in a minute. So here's some more cells, microvilli, folds, and the materials are going down the nephron like this. So this is the opposite side of the wall, microvilli, folds, next cell, microvilli, folds. So if you were to look at cells under the microscope of the proximal convoluted tubule, what you would see is these folds appear as kind of like we call it a brush border under the microscope. So you wouldn't see the individual microvilli with a microscope, but you would see them as a brush border. So if you've done microscope work recently on the kidney, then you'll remember seeing that. So I think 13D has seen microscope work from the microvilli, but 13E hasn't yet, but we will. And then you can be very excited when you see this microvilli-like structures, which are referred to as the brush border if you're using a microscope. So microvilli, okay. So we'll also need to put on the capillaries, which are in close contact with the nephron. So they're surrounding the nephron all over the place. So let's pop on that capillary. So the capillary cells are sat on the basement membrane. So here's our endothelial cells. And opposite side of the capillary cells, endothelial cells. And I shove in a red blood cell just because then it looks like a capillary. Could do the same on the opposite side. Let's just quickly do that then. Red blood cell going by. Endothelial cells. Okay. Opposite side, blah, blah, blah. Right, so we've got the situation then where we're going to have the filtrate travelling down that's containing all the good materials going through here, and all the stuff that you want to get rid of, stuff like urea, will be travelling off this way, and the stuff that you've also, all the good stuff, we want to then be able to reabsorb and put back into the bloodstream. So I'm just looking around. I'm just currently in my daughter's bedroom, seeing if she's got any coloured pens, because I've had to move out the front room because you might have heard the budgie noises a little bit earlier. Let's have a look what she's got. Right, we've got some colours. I don't know if these work. Let's have a go with these. Right, so first of all, let's learn a bit more about these cells. So a couple of things they have. Now, we mentioned the microvilli, and why they have microvilli is to create a very, very large surface area so that we can have lots of symports, co-transporters. So these co-transporters, which I'll put on here, these carrier proteins, are able to carry 2 molecules at a time. So what they're able to move into the cell is glucose or amino acids, but they can only work in the presence of sodium ions. So if I was to draw one of these blobs, which are our co-transporters, sort of blown up a little bit bigger, what you would see on there is that they have areas for transporting sodium ions, but also areas at the same time to bring in glucose. So glucose can attach to it, as does sodium, and the other ones, exactly the same idea, can transport sodiums and amino acids. So these co-transporters, which are just drawn as the black blobs, are on the microvilli of your proximal convoluted tubule cells. We also have in the cell mitochondria, because there's a very energetic process you'll be familiar with on the opposite side of the cell taking place. So on this side of the cell we have the sodium-potassium pump. So sodiums are going to be pumped out this way, potassium's brought in, and this needs ATP. And yes, you are right to ask the question, is it the same number as it is in a nerve? Yes, all these sodium-potassium pumps in the body work exactly the same way. So although your textbook just says there's a sodium-potassium pump, yes, you are right, they will be doing the 3-to-2. So it's not modified, they are the same pumps in all the cells. And occurring over here as well, so sodium's being pumped out, potassium's being pumped in, ATP. Now because they need ATP, that's why you have lots and lots of mitochondria in these cells of your proximal convoluted tubule. So mitochondria there to provide ATP so that we can have the sodium-potassium pump in operation. So let's have a look now at what goes on as the materials move through. So if we've got something like, say, water, does this pen work? So yeah, here's some water molecules. So if you've got water in your filtrate, and what we've got is quite a large amount of water, so we've got high water potential here compared to inside the cells, the water can move into those cells simply by osmosis. And as it moves in, you create a higher water potential, so then the water will just keep moving into the bloodstream. So water is going to be moving by osmosis. Nice. We also then have something like glucose. Let's see if this red pen works. So if glucose is in the bloodstream, then glucose will be taken in by the co-transporters. So it'll bond with the co-transporter, and at the same time sodiums will as well. So sodiums will also bond to the co-transporter, and they will be brought into the cell. So we'll have sodiums and we'll have some glucoses in. The other co-transporters work in the same way, is that we'd have some sodiums being brought in alongside with some, let's see, we've got a different colour down here. Let's see if this one works. So you'd also have some sodiums being brought in with, where's this one? With some amino acids. Looks like, yeah, it works. So the amino acids will be coming in too. So the co-transporters bring sodium in and amino acids, or sodium and glucose. Now once they're brought into the cell, they will then be moving through the cell, diffusing their way through, and then they'll be removed out of the cell by facilitated diffusion. So on the opposite side, there'll be your typical carrier proteins, which will allow our glucose will be diffusing its way into the bloodstream, because it'll be going from a high concentration to a low concentration. The same for the amino acids. So the amino acids will also diffuse into the bloodstream, and because they're going to have to go through a carrier protein, both of those molecules will be going by facilitated diffusion. So we have facilitated diffusion. That's for glucose and amino acids. Now to maintain this movement of materials of glucose and amino acids into the cell, they were moved in by this co-transporter. Now this only works if the sodium which is moving in is going from, say, a high concentration to a low concentration, and we maintain that concentration gradient by having this ATP pump at the other side, because as the sodium starts to build up inside the cell, they are pumped out into the bloodstream, and therefore you always maintain a concentration gradient of sodium ions across the cell. So what we've been able to remove is we've been able to reabsorb glucose by using a co-transporter. So that came in on the co-transporter along with sodium into the cell. Then, because it was in a high concentration inside the cell compared to the outside, the glucose was then be able to diffuse its way through. We know that glucose is a large molecule, so although we say diffusion, it's really facilitated diffusion, because it would need a carrier protein to help it move through the membrane. The same for amino acids. The amino acids were travelling in the bloodstream, and they got filtered into the filtrate. In the filtrate, you want to reabsorb them, so they will attach to the co-transporters along with sodium, which will bring them into the cell. They'll diffuse their way through to the carrier proteins, which will help them diffuse through into the bloodstream. So the amino acids are also moving by facilitated diffusion. We have our sodiums, which are moving into the cell by the co-transporter, and they're moved out by this active pump at this side. What else do we have? We have water molecules, which are moving in by osmosis. Now everything else, so things like urea, will just simply continue because we don't have any co-transporters to take urea back into the cell. So the urea molecules will just carry on into the filtrate and off onto the next stage of nephron. So at this stage, we've selectively reabsorbed about 85% of the materials which were filtered out originally are now reabsorbed. So I just want to see if I've got a bit of space to write some of that down again in case you wanted to have a bit of a summary. So what you've got in the proximal convoluted tubule, you have got selective reabsorption. So let's deal with, so glucose was dealt with by having a co-transporter to bring it into the cell, and then it relied on facilitated diffusion into the blood. Same for amino acids. So the amino acids, exactly the same idea, co-transporter that brought it out of the nephron into the proximal convoluted tubules. Then it relied on facilitated diffusion to then ultimately move it into the blood. So these processes are not active processes, but some people sometimes refer to them as being reliant on active transport. So sometimes they say it's secondary active transport, because actually if you didn't have that active pump working, then this whole process would shut down, because those co-transporters rely on sodium being present for the co-transporter to work. So if you can't then move sodiums out, then you can't get the co-transporter to work. So sometimes this is referred to as secondary active transport. Right, so glucose and amino acids are used that way. Water was quite straightforward actually, that was just osmosis, nice and straightforward, through the cells into the blood. And what was the tricky one? The tricky one was the sodium. So the sodium brought in by a co-transporter, but this would only work because we need to keep that concentration gradient, so it only works because you actually have the sodium potassium active pump in operation, moving the sodiums into the blood, and therefore the diffusion gradient will then work to allow the co-transporter to work. So glucose, amino acids, water and sodium have all been taken care of. Urea stays in the filtrate, it can't get out, it can't get into those proximal convoluted tubules. So what the stages we've just looked at there is, we've just looked at this stage here in the proximal convoluted tubule. So it's a good idea to stop at this stage, and we'll try out some exam questions before we go any further.